This is the aerobic metabolism clip. Just a reminder that aerobic metabolism occurs in the mitochondria of the cell. And you'll notice on the next few slides that I've broken the stages of aerobic metabolism into the first, second, third, and fourth stage. This is not something that you'll necessarily see in a textbook, but I thought this was the best way to separate the information so it would make the most sense. The first stage of the aerobic metabolism of a molecule of glucose is actually glycolysis. And this can be a little bit confusing because in the previous clip you learnt that glycolysis was the anaerobic breakdown of glucose. If we take a close look at this pathway, you'll see that there's one important difference between this pathway and the pathway I showed you in the previous clip. In the pathway in the previous clip, the very final stage is the formation of lactic acid from pyruvate. So the final step where lactic acid is produced is not part of this glycolytic pathway as part of aerobic metabolism. So in other words, the pathway stops one chemical reaction short of the anaerobic glycolysis pathway. The final net products for the first stage of the aerobic metabolism of glucose is 2 NADH, shown here, 2 ATP, and that's the same as anaerobic glycolysis, but instead of 2 lactic acid molecules, we have 2 pyruvic acid molecules, or 2 pyruvate molecules. So it's very similar, but there are some important differences. Key to this pathway is that the pyruvate molecules will go ahead and enter the mitochondria, and I'll show you this on the next slide. The NADH molecules also give up their hydrogens to the mitochondria. So on to the second stage of the aerobic breakdown of glucose. This occurs in the matrix of the mitochondria. Now, if we take a close look at the structure of the mitochondria, you see that there's an outer membrane shown here, but there's also an inner membrane. Okay, so what we actually have are two compartments within the mitochondria. We have the matrix, which is the inner compartment, and then we have another compartment in between the outer membrane and the inner membrane. And this does become important when we look at the different metabolic pathways occurring in the mitochondria. So if you remember back to the previous slide, the end products were 2 pyruvic acid molecules along with 2 ATP and 2 NADH. So these pyruvic acid molecules that are produced in the cytoplasm in glycolysis actually enter the matrix of the mitochondria. And you'll see here, this shows the chemical reaction that occurs. Pyruvate is converted into a molecule called acetyl-CoA. In that process, you get the production of a carbon dioxide molecule and the production of an NADH molecule. Remember that one glucose molecule entered glycolysis and 2 pyruvic acid molecules were produced. So this will happen times 2. So the final net products of the second stage are 2 acetyl-CoA, 2 carbon dioxide, and 2 NADH. Remembering that carbon dioxide is a waste product that will be exhaled via the respiratory system. Now on to the third stage of the aerobic metabolism of glucose. Remember that acetyl-CoA were final products in the previous slide. This acetyl-CoA is shown here, and the chemical reaction that I showed you on the previous slide is shown here. Acetyl-CoA enters what we call the Krebs cycle, and the Krebs cycle is shown here. And again, similar to what I've said before, every arrow on the slide indicates a chemical reaction. And each substance within these rectangular shapes represents the substrate and the products. And remember that the initial substrate is converted into a product, This then becomes the substrate for the next reaction, and so on. So we often say that the Krebs cycle is a cyclic pathway because, as you can see here from the diagram, it's circular. So in other words, the end product becomes a substrate again in the cycle. If we think about what the most important function is of the Krebs cycle, it's to complete the oxidation or hydrogen removal of acetyl-CoA. So remember that acetyl-CoA is the initial substrate, and remember back to previous clips, the production of ATP comes from hydrogen removal from originally those foodstuffs that contained, for example, in this case that we're looking at, glucose. So all along the way in these metabolic pathways, we're removing hydrogens, and as you'll see in the final stage, these hydrogens are really important for the eventual production of large amounts of ATP. So this is still on the third stage, so still on the Krebs cycle, but showing you the most important products and where they're formed. Remember that for one glucose molecule, you get the production of 2 pyruvate molecules, and therefore the production of 2 acetyl-CoA. So for every glucose that's broken down, you get 2 acetyl-CoA molecules. This means that you get 2 turns of the Krebs cycle for every individual glucose molecule. So for each turn of the cycle, there are 2 carbon dioxide molecules produced, shown here and here, 3 NADH, shown here, here, and here, 1 ATP, and 1 FADH. Because you have 2 turns of the cycle, the net products, or final products, are 4 carbon dioxide, 6 NADH, 2 ATP, and 2 FADH. Okay, so just to summarise, the most important steps in the Krebs cycle in terms of energy production, so in terms of ATP production, are the formation of NADH and FADH. And what's key with these molecules is the hydrogens that they're transporting. Remember that NADH and FADH are formed when NAD and FAD pick up hydrogen atoms, and that's shown here. So these hydrogen atoms are taken to another place in the mitochondria where the ATP is produced, and that's what I'll show you in the next stage. So if you think about the first 3 stages of the aerobic metabolism of glucose, the most important parts are the oxidation, the hydrogen removal of the substrates. And when the hydrogen's removed, you have the production of NADH and FADH. Even though ATP is produced in the Krebs cycle, you'll see that this is a very small amount compared to the ATP that can be generated from the NADH and the FADH. So on to the 4th stage, and this is where most of the ATP is actually produced. And the pathway is called the electron transport chain. It's given this name because of the nature of the pathway. The pathway is shown here. If we look a bit closer at this diagram, this is the outer membrane of the mitochondria. Okay, so this section here is blown up into this diagram here. This is the outer membrane of the mitochondria and the inner membrane. So in this region here is the matrix. So the matrix is where the pyruvate is broken down into acetyl-CoA, and the matrix is also where the Krebs cycle takes place. The electron transport chain takes place within this inner membrane of the mitochondria. These purple structures shown here are proteins called cytochromes, and their function is to pick up electrons from the hydrogens associated with the NADH and the FADH. Then the cytochromes transport those electrons along the electron transport chain. and energy is derived from the transport of these electrons, and it's used to phosphorylate ADP to form ATP. Phosphorylate may be a term that you haven't come across before. Basically, it means to add a phosphate group to a molecule. So when we add a phosphate group to ADP, we form ATP. So the next slide is really looking at what goes on in the electron transport chain in some more detail. So if we look at the first step, NAD and FAD give up their electrons to the electron transport chain, or in other words, give up their electrons to the cytochromes. We can see NADH here giving up 2 electrons. Remember that a hydrogen atom contains a proton and an electron. So for every NADH molecule, there are 2 hydrogens and therefore 2 electrons to give up to the cytochrome. FADH is shown here, also giving up 2 electrons to the electron transport chain. The electrons are moved along the chain, and as they do so, energy is used to pump the protons across this membrane to this outer compartment. So this is the space between the outer membrane and the inner membrane. And remember, this section here is the matrix. So the electrons being passed along provide the energy to pump the protons across into this outer compartment. A key point to note here is that these protons can't diffuse across the membrane. However, there is a region where there's a channel where they diffuse down their concentration gradient back into the matrix. This channel is associated with a complex protein shown here, and this is ATP synthase. Remember that ATP synthase is the enzyme that catalyzes the production of ATP from ADP and inorganic phosphate. A key point to note is that when these protons are diffusing through this channel, it activates the enzyme. And so that the function of that enzyme is to combine ADP and inorganic phosphate to form ATP. We all know that oxygen is important for metabolism, and after all, we are talking about aerobic metabolism where oxygen is used. Oxygen is very important in that it acts as what we call a final electron acceptor. So after these electrons have been passed down the cytochromes, the oxygen accepts the electrons and the protons that were associated with those electrons and therefore hydrogen atoms. So the oxygen accepts, in other words, the hydrogen atoms to form water, and that's where water is formed. Oxygen is absolutely essential for this process because if it's not there to accept those electrons that are flowing down the chain, movement down that chain, or I should say electron movement down that chain, will stop, and therefore ATP production will also stop. So in other words, electrons have to be constantly moving down this chain to provide the energy to pump the protons into this compartment. So they'll then diffuse down the channel, activate the enzyme which forms ATP from ADP and inorganic phosphate. And one final point, and it's shown here in point 4, is that ATP is transported out of the inner compartment by a carrier protein, because remember, we're located here within the mitochondria, and for muscle contraction, this ATP needs to get out into the cytoplasm and to the sarcomeres, where it can be broken down and support muscle contraction. So when the ATP is moved out via this carrier, it's exchanged for ADP and also PI. Two terms that you should be familiar with are direct or substrate level phosphorylation and oxidative phosphorylation. Direct or substrate level phosphorylation refers to the direct production of ATP in glycolysis and also the Krebs cycle. And you can see here, as I mentioned before, this is where an ATP is produced per cycle of the Krebs cycle. So 2 of these are produced per glucose molecule. This is direct or substrate level phosphorylation. This is different from oxidative phosphorylation, which is the ATP formation that occurs in the electron transport chain. It's referred to as oxidative phosphorylation because you have a series of oxidation reduction reactions in the electron transport chain that results in the eventual production of ATP. Now if we look at the tally from an individual molecule of glucose broken down aerobically, you'll see a grand total of 32 ATP. Remember that there were 2 ATP produced by glycolysis. So the aerobic breakdown of glucose produces a much larger amount of ATP than the anaerobic breakdown of glucose. The overall efficiency of aerobic respiration is at most 20 to 30%. And I did mention this point earlier in a previous clip. So that means that 70 to 80% of energy is released as heat. What's shown here in this table is, along with the tally of 32 ATP, where the ATP come from. So if we start with glycolysis, and remember this is not anaerobic glycolysis, it's glycolysis as the first stage in the aerobic breakdown of glucose, you still gain that 2 ATP for the cell that's produced in glycolysis, but you also have the production of 2 NADH. If we look at the key down the bottom of the slide here, it's showing you that for every NADH molecule, there's a yield of 2.5 ATP. And for every FADH molecule, there's a yield of 1.5 ATP. Now, it's not possible to have half an ATP molecule, but this is the gain for the cell. There is an energy cost of getting the ATP out into the cytoplasm, so this explains the half allocated ATP. So given that for every NADH molecule you get 2.5 ATP, the tally there is 5 ATP. The next stage was the conversion of pyruvic acid into acetyl-CoA. And remember that 2 pyruvic acid molecules were produced from the 1 original glucose molecule. 2 NADH were produced, which again gives you 5 ATP. Within the Krebs cycle, again remember that 2 pyruvic acid were converted into 2 acetyl-CoA, which entered the Krebs cycle. So there were 2 turns of the Krebs cycle for the original glucose molecule. This 2 GTP refers to the direct production of ATP within the Krebs cycle. 6 NADH and 2 FADH are also produced, giving 15 and 3 ATP. If you add up all of these values, you get the total net gain for the cell of 32 ATP. It's suggested now that you watch the animation on how the Krebs cycle works. And again, this will show you in animation form what I was describing on previous slides, and also an animation on the electron transport system. If there's any additional detail in these videos, then you're not responsible for that additional information in exams. These videos are not supposed to show you new information, but actually support the video clips that you've already been shown. So it's recommended that you attempt the quiz on LMS called Aerobic Metabolism, and when you feel like you've mastered this topic, you can continue on with the clip Glucose and Energy.